Hello, everyone. My name is Brennen and I will be discussing drugs of abuse affecting neurodevelopment and regenerating Schmidtea Mediterranea planaria on behalf of the Staley Lab at Rowan University. So the objective of this was to evaluate and compare the effects of ethanol on neuroregeneration by introducing ethanol before and after resection of these planaria worms that you can see on our right. Schmidtea Mediterranea planaria are widely known for being the simplest organism with a central nervous system, and they have a regenerative ability. So basically, if you cut these worms into three different pieces, as you can see on the bottom, they'll regrow what they're missing. Previous studies have used head resection as an effective, inexpensive model for in vivo animal experiments for the effects of various chemicals on neurodevelopment. And then by using their evolutionary aversion to light, it's possible to model the effects of drugs of abuse through changes in their light avoidance behavior, which we can quantify through the planaria photophobic behavioral index. So to test this, we had 80 worms that we separated into two groups. 40 worms are exposed to control water and 40 worms are exposed to 1% ethanol. And then after 48 hours, all the worms will be decapitated, and the two previous groups are then divided into four total groups. A group will be exposed to control in the pretest and then control during the actual video tests. And then we have the other groups of control to ethanol, ethanol to control, and ethanol to ethanol. And the pods you can see at the bottom are what these houses are going to be tested in. So we put these pods in our high throughput machine, which you can see in the picture below. It consists of the tray, the housing container, the light, and the cap. And this basically allows us to expose the worms to light without any risk of outside contamination. So we prime these worms by shining light in the middle of the pods, basically forcing all the worms to the dark of the outside. And then after 60 seconds, we switch the lights so the outside is now lit, and we record them for 90 seconds and track them swimming back to the darkness towards the inside. We then can analyze these videos through our studio in Fiji. This is the data that we got from that high throughput testing. And the most interesting parts is that regardless of the initial exposure conditions, worms in ethanol had a significantly reduced PPBI score compared to those control. And then over the course of 8 days, all exposure conditions except for the full ethanol exposure trended towards increased photophobic behavior. This makes sense because we do know that ethanol causes a delay in regeneration. Peculiar, however, in the planaria tested in control water, those exposed to ethanol prior to testing actually had a more photophobic behavior than control. We're not entirely sure why this happened, but this could be due to the fact that we changed it from a highly toxic environment to a non-toxic normal environment, which kind of shocked it into more activity. In the future, we hope to evaluate the effects of ethanol on the physical nervous system through immunohistochemistry, and we also hope to test this experiment with other drugs of abuse to evaluate the scope of planaria as a model. Thank you.